students. This is Dr. Shabana Begum, Associate Professor in Zoology, Maharani Science College for Women, Bangalore. Today I am going to discuss on the topic germ layers and diploblastic and triploblastic conditions in animals, which is one of the subtopic under BSc first semester Unit 1, that is Animal Architecture. In my previous class, I have discussed about the subtopic called body organization, and you have learnt 5 different types of body organizations, such as the protoplasmic type of body organization. Example is amoeba. Cellular level of body organization, the example is sponges. Then tissue level or grade of body organization, example the coelentrates. Then organ level of body organization, the example being the platyhelminthes forms, and organ system grade or level of organization are the aschelminthes forms to mammals, that is higher invertebrates up to chordates exhibit organ system level of body organization. Today let us learn about an interesting subtopic called germ layers. As you all know, germ layers are the embryonic tissue layers that are formed during the process of gastrulation in the early embryonic life of an organism. So, based on the number of germ layers developed in the embryonic stage, animals are classified into 2 types, that is diploblastic and triploblastic. So, here the learning objective of this topic being to know about the germ layers, then to differentiate diploblastic and triploblastic organization. The schematic diagram in this particular slide indicate the diploblastic and triploblastic condition in organisms. The introduction to this topic, see here, all animals develop their tissues, organs and organ system from the cells that form the embryonic layers. So, based on the number of embryonic layers, animals are classified into 2 that I just mentioned, that is the diploblastic and triploblastic animals. Different organisms have different cell layers in the embryonic stages that form various organs eventually. In metazoa, special regions of the body are set aside for dealing with different functions. The metazoans produce gametes of 2 types. The male gametes are the spermatozoa and the female gametes are the ova. A spermatozoon fertilizes an ovum to form a zygote, which undergoes a series of mitotic cell divisions called cleavage divisions to form a hollow ball of cells called blastula, which has a cavity known as blastocele. Further, increase of the cells of the blastula causes an invagination of the wall on one side, and by different processes that they are called morphogenetic movements, the cells eventually come to lie in 2 layers, an outer layer of ectoderm and an inner layer of endoderm. The blastocele is obliterated. The mouth of the invagination is a blastopore, which leads into a new cavity called the archenteron. This 2-layered bag-like structure is a gastrula. So, this slide with the figures depict the early embryonic development in animals, starting from the union of male and female gamete forming a diploid zygote. The zygote will undergo cleavage divisions. Here only one cleavage divisions, that is cleavage pattern called 8 cell stage is shown. Then that ultimately will form a blastula stage, which in cross section is shown to contain a cavity called as blastocele, which further undergo certain morphogenetic movements and form what is known as a gastrula, ultimately with the 2 or 3 primary germ layers called ectoderm, endoderm or mesoderm. So, the formation goes like this. So, zygote of an animal undergoes successive mitotic cell divisions called cleavage divisions. Only one cell, that is a cleavage stage here, the 8-cell embryo is shown in this diagram, which leads to the formation of gastrula. So, in most animals, cleavage results in the formation of a multicellular stage called a blastula. So, the blastula of many animals is a hollow ball of cells. Most animals also undergo gastrulation. Here, there is a rearrangement of the embryo in which one end of the embryo folds inward, that is called the invagination. Then it expands and eventually fills the blastocele, and that gets obliterated, producing layers of embryonic tissue, the ectoderm, which is the outer layer, and the endoderm, called the inner layer. Then the blind pouch formed by gastrulation is called the archenteron, which opens to the outside via the blastopore. The endoderm of the archenteron develops into the tissues which line the animal's digestive tract. Another information here, the development of some metazoans stops at gastrula stage. These two-layered metazoa are diploblastic, such as Cnidaria and Ctenophora. In all other metazoan phyla, a third layer of cells called mesoderm arises between the ectoderm and endoderm. The phyla which possess 3 layers are triploblastic, and their mesoderm opens up further possibilities of increase in size and complexity. The diploblastic animals. Here, when the cells are arranged in 2 embryonic layers, the animal is called a diploblastic animal. The 2 layers are the primary germ layers called ectoderm and endoderm. Then mesoglia, which is an undifferentiated layer, is present between the ectoderm and endoderm. So, diploblastic animals possess radial symmetry. Example, Cnidaria and Ctenophora, and they are considered as diploblastic. Then jellyfishes, comb jellies, corals and sea anemones are the other examples of diploblastic animals. So, this figure in the slide will depict the 3 germ layers, especially the outer ectoderm, inner endoderm, and in between which there is a jelly-like mesoglia present in case of diploblastic animals. There are 2 types of diploblastic conditions in animals, that is the apparent diploblasty and absolute diploblasty. So, apparent diploblasty, here examples are sponges. So, sponges have a cellular level of organization, and hence ectoderm and endoderm terms are not there. Instead, the body wall consists of 2 layers, an outer protective layer called pinacoderm formed from pinacocytes and porocyte cells, and an inner guanoderm consists of flagellated guanocytes or collar cells, which are nutritive and reproductive in function, with a non-cellular mesenchyme in between. It consists of amoeboid cells, scleroblast cells, totipotent archaeocytes, etc. There are no sensory or nerve cells. As a result, they are not truly diploblastic. Examples are the poriferans. So, what is the significance of this type of diploblastic? Here, first point you can see, the aggregation of cells to form body layers increases the capacity of the body to resist physical stress of the environment. Next point is the protozoan characters. In sponges include cellular independence in their bodies as in colonial protozoa. Then totipotent nature of their cells, absence of body organs or tissue level organization, absence of gonads, and digestion being intracellular by endophagy. There also have collar cells and amoeboid cells as found in colonial guanoflagellates. And what is absolute diploblasty? This is the second type of diploblastic condition. Examples are the coelentrates. So, coelentrates are truly diploblastic. in which the body is made up of two distinct tissue layers developed from the ectoderm and endoderm, while mesoderm is absent. Mesoglia, a jelly-like substance present between the two layers, contains fibrous connective tissue and amoeboid cells. The outer layer is called the epidermis and is composed of epitheliomuscular cells, gland cells, cnidoblast cells, interstitial cells, sensory cells, nerve cells and germ cells. The inner layer is called the gastrodermis. It is composed of nutritive cells, gland cells, sensory cells, nerve cells and muscular cells which develop from ectoderm and endoderm. What's the significance of this absolute diploblastic conditions? Here, the first point you can mention, the two layers by having different cells function more independently and efficiently with clear division of labor. There, second point, the presence of nerve cells forming nerve net brings about better coordination of cells, hence they form tissues. Next type is triploblastic condition or triploblasty or triploblastic animals. So, when the cells are arranged in three embryonic layers, which are the germ layers or primary germ layers, the animal is called triploblastic animal. So, the three layers are the ectoderm, mesoderm and endoderm. Animals belonging to phyla Platyhelminthes to Chordata have a triploblastic organization. So, they belong to the class Lateria and possess bilateral symmetry. So, the figures here indicate the conditions of diploblasty and triploblasty in organisms. You can clearly see the arrangement of cells to form the ectoderm and endoderm along with mesoglia in diploblastic condition, and the condition where the ectoderm, mesoderm and endoderm germ layers are seen is the triploblastic condition. So, here the triploblastic animals are further divided into sections such as acoelomates, like they lack coelom, eucoelomates which have true coelom and pseudocoelomates which have false coelom. So, eucoelomates are again divided into two sections based on the position of blastopore and what it develops into. For example, in protostomes, the mouth develops from blastopore, whereas in deuterostomes, the anus developed from the blastopore. So, it is believed that diploblastic animals gave rise to triploblastic animals around 580 to 650 years ago. So, here are the pictures of acoelomate, eucoelomate and pseudocoelomate condition. So, in triploblastic phyla, the ectoderm and endoderm retain most of the function which they perform in diploblastic animals. The ectoderm forms the outer protective epidermis, external sense organs, nephridia and the nervous system. But in case of echinodermata, part of the nervous system is mesodermal in origin. The endoderm gives rise to the lining of alimentary canal and organs associated with digestion and respiration, while the mesoderm is not single entity but has parts which originate in two ways. The cells which migrate from the ectoderm or endoderm form a loose cellular tissue called mesenchyme which fills the spaces between the outer layers, and it is comparable to the cells which are found in the mesoglia or cnidaria. The second type of cells which form the wall of the body cavity are known as mesothelium or real mesoderm, which gives rise to connective tissue, muscles, skeleton, blood, circulatory system, excretory system and reproductive system. In lower triploblastic phyla, that is the Platyhelminthes and Aschelminthes, there is no mesothelium, but the other phyla possess both kinds of mesoderm. The triploblastic acoelomate animals may attain a degree of complexity which is not seen in diploblastic animals. So, the differences between diploblastic and triploblastic animals, in the form of their definitions, symmetry, mesodermal development, body cavity is mentioned here. You please go through the slide. So, at the end of my lecture, the outcome of the study is here. The animals like sea sponges exhibit the simplest organization consisting only a single germ layer. Though they are composed of differentiated cells, they lack true tissue coordination. Diploblastic animals, on the other hand, exhibit an increased complexity than the sponges, containing two germ layers, the ectoderm and endoderm. They are organized into recognizable tissues. Triploblastic animals possess an additional germ layer, the mesoderm, from which they can develop complex organs in the body. Thus, the key difference between diploblastic and triploblastic animals is the type of the cleavage during embryonic development. So, at the end of the lecture, some MCQs, that is multiple choice questions, you can answer to know whether you have understood the topic or not. The first question is, the body of which diploblastic animal are divided from only two, is derived from only two embryonic cell layers, that is ectoderm and endoderm, but no mesoderm. The answer is A, sponges and coelentrates, B, protozoans, C, microbes and D, none. So, the answer being the option A, that is sponges and coelentrates. Second question, common characteristics of coelentrates and sponges are A, triploblastic and pseudocoelomate, B, triploblastic and acoelomate, C, diploblastic and acoelomate and D is monoblastic and acoelomate. The answer is option C, diploblastic and acoelomate. Third MCQ, which of the following statements is true about triploblastic animals? Options are, animals have two layers of cells which differentiate into tissue. Animals have three layers of cells which differentiate into tissue. C is, animals have three layers of cells which cannot differentiate into tissue. And D is, animals have four layers of cells which differentiate into tissue. So, the answer is answer B, animals which have three layers of cells which get differentiated into tissues. So, the next question. So, Platyhelminthes are best described as A, flatworms, triploblastic and acoelomate animals, B, flatworms, diploblastic and acoelomates, C, flatworms, triploblastic, coelomates, D is flatworms, triploblastic and pseudocoelomate animals. So, answer is the flatworms, the triploblastic and acoelomate animals, that is option A. So, Platyhelminthes are best described as the flatworms, triploblastic and acoelomate animals. Next question, diploblastic animals occur in A, phylum Annelida, B, phylum Porifera, C, phylum Coelentrata and D, both B and C. So, the answer being the option D, that is both B and C, that is the diploblastic animals occur in phylum Porifera and Coelentrata. So, here are few references. You can always go through them in case if you want to further study more detailed